I'm happy to invite Professor Dr. Alexey Stepanov, who is the head of the Department of Soil Biology in Moscow State University, Faculty of Soil Science.
Dear colleagues, first of all, I would like to thank Mikhail Semyonov for the possibility to speak here. It's very important for me. My presentation concerns bactohumic substances as a basis of nature-like technologies for soil fertility and plant growth. Humic substances are widely used to increase soil fertility and plant production in polluted air and soil remediation purposes. Humic substances, because humic substances has a high biological activity, they are resistant to microbial degradation in the soil. They has a natural organic polymer, so no negative effect on environmental. So that impact of humic substances on soil fertility can be direct by changes in the physical-chemical soil parameters and indirectly by changes of microbial activity. With regards to living organisms in the soil are especially important protective and physiological function of humic substances. So, how microorganisms are distributed in the soil? They developed in the films of humus on the surface of mineral. So, variants of localization of microbial cells on the surface of the organic mineral particles are different, and cells' microbial activity depends on the type of contact. So our approach is to absorb humic substances on the surface of microbial cells as like in soil, in the natural environment, and create bactohumic preparation, BHP. For these purposes, we should isolate a microbial culture which use humic substances as a source of carbon and energy. We should select a non-toxic concentration of humic substances for development of microorganisms. We should control the number of microbial cells in the processes of growth on humic substances media, and we should check stability of the humic substances preparation in aqueous media. Here you can see absorption of humic substances on the microbial cell surfaces using electronic microscope. Humic substances adsorbed on the outer membrane of gram-negative or cell wall in gram-positive microorganism create the net for freely permeable elements in mineral nutrition and molecules of organic components for microbial cells such as sugars or amino acids, and at the same time toxic compounds such as heavy metals or other toxins in environmental condition bind by humic substances, and this character of interaction of humic substances with microbial cells will have a protective effect on microbial cells in environment, in soil, for example, and to promote their survival in and increased activity in soil. So, interaction of humic substances and microorganisms based on microbial utilizing of adsorbed humic substances as energy sources. Humic substances stimulate activity of microbial cells and microorganisms, and the bacteria create a stable bactohumic products. Our work was divided by two stages. In the first stage, our work was carried out with solution of potassium humate and pure culture of bacteria of the genera Rhodococcus, Azotobacter and Pseudomonas, as well as native microbial complex isolated from organic clay soil in Moscow, from Moscow region. Experiments on remediation of soil from petroleum products and heavy metal were carried in podzolic soil. Oil, diesel fuel and water-soluble metal salts were introduced as a pollutant in five-fold excess of the maximum permissional concentration, and plant of cress salad were used as a test culture. So, our experiments show that the bacteria can survive in the form of bactohumic preparation in water solution not less than 1 year at high level of bacterial number in process of storage as bacterial humic preparation. Bactohumic preparation influence on the structure of the complex of soil microbial in contaminated soil by, for example, by heavy metals. You can see that polluted air, structure of microbial community in polluted soil, very different compared with control. But after treatment of bactohumic preparation, the structure of bacterial component of soil reached to control level. And bactohumic preparation influence on the growth and development of plant in polluted soil by, first of all, by heavy metals. You can see that the biomass, dry weight biomass of plants of cress salad reached to control. But visually it is very interesting. It's the control number 1 and polluted variant number 4. You can see the difference in the development of plants, and after treatment of bactohumic preparation, you can see that it's not visible difference between control and polluted and growth of plant in polluted soil. Very interesting that bactohumic preparation can production of plant production in polluted soil by crude oil and diesel fuel has a positive effect, but you can see the number 6, the concentration of lignogumat is very high, and in such a high concentration of humic substances is toxic for microorganisms. It's a very important point. So benefits of bactohumic product is increasing the storage time of the products as a form of bactohumic preparation, the increase of resistant time in environmental, high level of biological activity, and the possibility of use in microbial cells in unfavorable environmental condition, such in soil salinity and in high concentration of heavy metals, and in soil polluted by oil. In the next stage, we carried out joint investigation with our colleagues from Udayana University from Indonesia. We use indicator plant as a cabbage, Brassica oleracea, which is susceptible to root disease in a field experiment. You can see healthy roots and club roots. We can see broken xylema. And the treatment was a variation of the concentration of an aqueous solution of lignogumat and trichoderma and spore of trichoderma asperillum isolated from cabbage plantation by our colleagues from Udayana University. You can see the total club roots exhibit of the cabbage root on lignogumat and trichoderma treatment, positive results of such kind of treatment, positive results in disease incidence of club roots on cabbage under treatment of lignogumat and trichoderma. Canopy dry weight and the lignogumat concentration is this variant is very useful for our investigation, and leaf area and after treatment of lignogumat is more positive. So total microbial exhibit and in the cabbage media culture growth of microbial population in bacteria, in fungi, and trichoderma after treatment of Bactohumic preparation. So application of Bactohumic preparation based on lignogumat and trichoderma lead to increase the biomass of cabbage, showed the lowest percentage of diseased plants and the smallest number of affected roots. So we can conclude that sorption of humic acid on the surface of bacterial cells was detected using electron microscope. The number of microorganisms in preparation of humic substances is stabilized at high level and stabilized for at least 1 year. Application of Bactohumics preparation increase the biological activity of soil and increase the biomass of plant in contaminated soil by heavy metals and petroleum products. And it was found the ability of preserve target population of bacteria in the native soil for several months in the case of their introduction as a form of Bactohumic preparation, and combination based on lignogumat and trichoderma in some cases make it possible to neutralize the toxic effect of phytopathogens. Thank you very much for your attention.
Yes, okay. Thank you. Maybe several questions for Alexey Stepanov. Do you have one?
This is a very good, very interesting work. I have one question. Yeah, the humic acid. Yeah, you use this. How this substance influence the microbe? Because before we also used some like amino acid. We found the one kind of amino acid can attract the bacillus, and also is benefit the colonization of the bacillus on the root surface. If you did some research about the humic acid, if they are influence the colonization, have some function in the biocontrol.
Yes, we can use humic substances for such purposes, and humic substances is a natural product, so there is many compounds which includes in humic substances. And sometimes humic substances has amino acids in compounds. Humic substances usually formed net in the surface of microbial cells so that they can protect microbial cells of negative influence of environments. And they can use for such purpose that you asked me. Well,
Thank you.
thank you.
Yes, I also have a question. How to obtain these substances? I mean that before use it in these bactohumic preparations, what is the technology of addition the humic substances on cells, and how to synthesize these humic substances?
That is know-how. So, but we can see some stages of make this kind of preparation. That one. First of all, you can isolate microbial culture, which can use humic substances as a source of carbon and energy. After that, you can select non-toxic concentration of humic substances, because in high concentration of humic substances, humic substances is toxic for development of microbial cells and plants. After that, you can control the number of microbial cells during the growth process on humic substances, and check stability of humic preparation in aqueous media. And after that, you can choose combination of humic substances and pure culture. It's a different.
How to get humic substances first? I mean that, yes, you wrote that the stage of selection of non-toxic concentrations of humic substances. This is the second, yes. But how to get it first? I mean the humic substances before.
You can buy humic substances as in the market. There is a special preparation, but pure humic substances you can buy, or you can isolate from soil.
How to isolate it from soil? Yes.
You can take soil. You can treatment soil by alkaline substance, and after you can such products you can treatment by acids, and you have acidic sediment, and you after that you can work with such kind of sediment if you want to isolate humic substances from soil, or you can buy it on market. Bayer, for example, corporation produce pure humic substances. You can buy it and check your microbial complex isolated from soil by use of trademark of humic substances.
Thank you very much. Возможно, у кого-то еще есть вопросы из коллег, кто вот сейчас у нас находится в Zoom. Кажется, вопросов нет. Спасибо большое еще раз, Алексей Львович.
Thank you.
Спасибо большое.